Hi students, today we are going to discuss about one more important short notes from lipid metabolism. The topic is apolipoprotein, right? Lipoprotein is different. I have uploaded separate videos for classification of lipoprotein. In the lipoprotein, the types are chylomicron, LDL cholesterol, HDL cholesterol, like that. For that also I have uploaded separate videos. At your free time, all of you go through the lipoprotein videos. Uh, very clearly, very elaborately about the lipoprotein metabolism as well as role of apolipoproteins in lipoprotein metabolism, right? So apolipoproteins, first you have to know the point, note the point for types, that is A, B, C, E and 1, 2, 3, 4. We have A, B, C, E, these letters for the types, and in this A we have 1, 2 like that Roman letters, right? So what are all the characters for this lipoprotein, apolipoprotein? Don't get confused with lipoprotein with apolipoprotein. Apolipoprotein is totally different. It is a protein component of lipoprotein, right? Second one is it only regulates the lipid transportation in plasma as well as uptake of tissues. These two role done by this apolipoprotein. It is usually synthesized from liver and intestine. Only two organs for apolipoproteins, mostly liver. This apolipoprotein has some special characters, like it acts as receptors in some areas. It acts as cofactors in for some enzymatic reactions, right? Receptors, we have three, that is apolipoprotein ApoA, B100 and ApoE. These three receives the cholesterol component, either LDL or lipoprotein component, either LDL or HDL or chylomicron. ApoA is act as a ligand for HDL cholesterol. ApoB100 and ApoE act as a ligand for LDL receptor, whereas ApoE act as a ligand for chylomicron. With the help of this only, it will get transported from one place to another. Coming to cofactors, some apolipoproteins either stimulate or inhibit the enzymatic reactions. We have two enzymes in lipoprotein metabolism common as LCAT, that is lecithin cholesterol acyl transferase, and one more is LPL, means lipoprotein lipase. So here ApoA1 increases the LCAT activity, whereas ApoA2 decreases LCAT activity, and ApoC2 involved in lipoprotein lipase enzyme reactions. Lipoprotein lipase reactions for ApoC2. These are all the general outline for your 5-mark short notes. Now we are coming to the types. In A we are having two types, A1 and A2, right? ApoA1, ApoA2. For this, you have to just mention 3 points, minimum 3 points. One is where it is synthesized from, usually liver or intestine. This ApoA1 is synthesized from both liver and intestine. The associated lipoprotein, that is, with the help of this apolipoprotein only, the lipoprotein will produce or synthesize from the liver or intestine, that is cholesterol, phospholipid, cholesterol ester, triacylglycerol with some apolipoprotein, 1 or 2, sometimes 3. So the associated lipoprotein, second point you have to mention for all the types, is chylomicron and HDL. So intestine means we know it is synthesized from intestine, chylomicron. HDL is synthesized from liver. That is why the associated lipoprotein is chylomicron and HDL. Functions, it has important function, that is ligand for HDL receptor, that is RCT, reverse cholesterol transport. This LCAT activator means lecithin cholesterol acyl transferase activator. This HDL and LCAT involved in reverse cholesterol transport. You know all very well about the reverse cholesterol transport. That is, from the peripheral tissues to the liver, the cholesterol is transported by LCAT enzyme. That is why it is anti-atherogenic lipoprotein, or apolipoprotein. Which one? ApoA1 is anti-atherogenic apolipoprotein. Why? It involves reverse cholesterol transport. What is reverse cholesterol transport? From the peripheries to the cholesterol, to the liver, the cholesterol is transported with the help of LCAT. That is reverse cholesterol transport. Second one is ApoA2. It is synthesized from liver. The associated lipoproteins, the same chylomicron and HDL. But here the action is LCAT inhibitor. This A1 is stimulator, whereas A2 is inhibitor. One more point for this MCQ point for this A1 is component of HDL2 and A2 is component of HDL3. We all know the HDL synthesis in first one is nascent HDL, second is HDL3, and finally HDL2 enters into the liver. So in this HDL2, the component is A1, whereas HDL3 component is A2. Very, very important MCQ. And coming to the B, we have again two types, B100 and B48. The number indicates 100 means 100% full size of the apolipoprotein, whereas 48 means B48 means 48% of the size of the B100. Right? They have different action. Usually synthesized from liver, associated lipoprotein, all the LDL group, VLDL, IDL and LDL. So it is a major lipoprotein of LDL. Which one? B100. What is the function of this? We have two functions. It binds LDL receptors. That is, receptor mediated endocytosis is the action mechanism in LDL receptor, LDL cholesterol. This receptor mediated endocytosis, the apolipoprotein is B100. This only receives the LDL cholesterol from the peripheral cells. That is why uptake of cholesterol from LDL given by peripheral cells. Right? Next one is B48. This is actually synthesized from intestine. That is a differentiating point between B100 and B48. B100 synthesized from liver, whereas B48 synthesized from intestine. Here the associated lipoprotein is only chylomicron. So it is a major apolipoprotein present in chylomicron. The function is formation and secretion of chylomicron from the intestine. So it is mostly related to intestine. It is mostly related to LDL cholesterol. Okay. Yes, the next type is ApoC1. That is ApoC. In C we are having three types, C1, C2, C3. All the three are synthesized from liver. All the three associated lipoproteins are chylomicron, VLDL and HDL. That means it involved in all the three components of lipoprotein. Coming to the function, C1 is different, C2, C3 is different. C1 is act on LCAT, whereas C2, C3 act on LPL. You should know the abbreviation of LCAT and LPL. LPL means lipoprotein lipase. LCAT means lecithin cholesterol acyl transferase, one of the important enzymes involved in HDL cholesterol metabolism. Okay. So here, ApoC1 activates the LCAT. Okay. And C2, C2 is stimulate the LPL enzymatic reactions, whereas C3 decreases, inhibiting the lipoprotein lipase reactions. Very, very important MCQ point. Coming to E, we have four types, but the 3 is not significant. The fourth one is very, very important because it involves in the development of CNS disease, Alzheimer's. Very, very important MCQ. Generally, ApoE is involved in kidney diseases like glomerulopathy and nephropathy. The main function of this ApoE is increases the uptake of chylomicron and LDL metabolism. That is remnants. Receptors are remnants. Another important question is, this is only apolipoprotein which is having rich amino acid, that is arginine, very very important MC. And here it is synthesized from again liver. Associated lipoproteins are again chylomicron, VLDL and HDL. So like that, you have to mention minimum 3 points, maximum 4 or 5 points for each and every type of apolipoprotein. It is very very important. Short notes from lipid metabolism. Okay.